Greetings to everyone. Welcome back once again to my educational channel on biology. I'm Teacher Janet, and today we'll be discussing an important topic, that is differences between mitosis and meiosis. So mitosis versus meiosis. What are the differences? So let's shine a spotlight on the differences because many students tend to get confused between these two types of cell divisions. Well, don't worry. After this lesson, you should be able to differentiate these two processes very well. Let's get started. Firstly, let's recap and review the definitions for mitosis and meiosis based on what is found in the textbook. So always give explanations that are based mainly on the textbook, unless the answer is not found there. Now, let's look at the definition for mitosis. Mitosis is the division of the nucleus of a parent cell into two nuclei. All right, two new nuclei with one nucleus in each daughter cell. That means two daughter cells are formed from one parent cell. The nucleus of each daughter cell contains the same number of chromosomes and the same genetic content as the nucleus of the parent cell. Okay, so we can say the nucleus of the daughter cell is genetically identical to that of the parent cell in terms of the number of chromosomes and the genetic content. So daughter cell and parent cell have the same genes. Now, example of mitosis. Mitosis occurs in the growth of a human being from zygote stage, which is the fertilized egg stage, to embryo, and then to fetus, baby, and adulthood, until through adulthood also. Okay, now meiosis, it is a process of nuclear division. So textbook use the word cell, but actually it's nuclear division, just like mitosis, that occurs in reproductive organs. So it occurs only in one part of the body, that is the reproductive organs, such as the testes and the ovaries, to produce gametes, specifically gametes which are reproductive cells, that contain half the number of chromosomes. That means the gametes have a haploid condition of the parent cell, which are diploid, right? So this is very important that we state that the gametes or the daughter cells formed in meiosis have only half the number of chromosomes compared to the parent cell. For humans, it will be 23 chromosomes in the gametes as compared to the 46 chromosomes in the parent cell. Okay, parent cell will have 46 chromosomes, gametes will have 23 chromosomes. Now, example of meiosis. It occurs in the testes and ovary, that means the reproductive organs, to produce the sperm and the ovum or ova. To produce sperms and ova. Ova is the plural form of ovum. Let's look at this very important question. Compare and contrast between mitosis and meiosis, 10 marks. So this question on the comparison between mitosis and meiosis can be asked in the SPM, in the essay question, structured question, or even objective questions, right? So let's find out the similarities and the differences. When we say the word compare, we have to mention or state some similarities and some differences. Now, there are two similarities here between mitosis and meiosis. Both processes involve cell division, in which a parent cell divides to form daughter cells. Okay, so mitosis and meiosis are actually nuclear division of the nucleus, all right, followed by cytokinesis, and these two processes, division of the nucleus and cytokinesis, makes up the cell division. Now, in both mitosis and meiosis, DNA replication occurs only once during the S phase of interphase. So to recap, this is the cell cycle. This diagram shows the cell cycle, which is divided into interphase and the M phase. So during interphase, the cell undergoes some some preparation before cell division. Okay, so there's G1, S, and G2 phase in interphase. And after that, there'll be M phase. It can be either mitosis or meiosis. And then the last part is the cytokinesis. The last phase is cytokinesis. Now, in the S phase, which is the second phase in the cell cycle, DNA replication occurs. So each chromosome will duplicate to form two sister chromatids. And in the chromosome, there is a strand of DNA. So actually what happens in the duplication of chromosomes is that the strand of DNA will make another copy of itself, or the cell will make another copy of this strand of DNA. Alright? So the two strands will be held together by the centromere. Right? So when the DNA, when there's another copy of DNA made by the cell, it's called replication of DNA. Alright? As a comparison, it's like you take a set of notes and photostat it. Alright? To make a photostatic copy of your original notes. So each chromosome will become two sister chromatids, and the DNA mass will be doubled, right? From the original DNA mass here to here, because you have two strands of DNA here now, the red strand and the pink strand. Okay, so the DNA mass has doubled. However, in terms of the number of chromosomes, it's still the same. This is one chromosome, and this is still considered one chromosome here after replication of DNA. Alright, not two chromosomes. One chromosome, but with the DNA mass doubled. So this replication of DNA or duplication of chromosomes occurs only once during the S phase of interphase before mitosis and meiosis starts. So the first difference is from the aspect of location. Where does mitosis occur? It occurs in the somatic cells in many body parts in a human, and especially, for example, in the skin. So there is a layer in the skin, the third layer from the top, called the Malpighian layer, where cells are constantly dividing by mitosis to produce new skin cells to replace the dead cells that have dropped off, right? And also in the bone marrow, blood cells are produced. For example, red blood cells, white blood cells, and platelets are produced to replace those blood cells that have died, that is in the blood, right? So the bone marrow is involved in producing new cells through mitosis to replace the dead blood cells. Now, how about the location of meiosis? It occurs only in cells in the reproductive organs or gonads. Gonads are the reproductive organs, which are the testes in males and ovary in females. So the location for the process of meiosis is quite limited. That is only in the reproductive organs, not in all parts of the body, right? So we can see here, this part here, the cross section of the testes, and the process of meiosis occurs inside the tubules here, small little tubes in the testes, called the seminiferous tubules, which we'll study later on in another chapter. For females, meiosis occurs in the ovaries. So females have a pair of ovaries, and and inside the ovary, meiosis occurs as the ovum develops slowly. Alright, so again, we'll be studying more of this in another chapter. Second difference is what type of cell is produced in mitosis and meiosis. So in mitosis, the type of cell produced are diploid somatic cells, such as red blood cells, muscle cells, nerve cells, intestinal cells, cells of the liver, and so forth. Now, what do we mean by diploid? A diploid cell is a cell that has two sets of chromosomes. One set is from the father and one set from the mother. So if we refer to a human being, in a diploid cell of a human being, there are 23 different chromosomes from the father of the person and 23 different chromosomes from the mother of the person. All right? So when they are found together in one cell, that cell is called a diploid cell. We use the term 2N, meaning the N here means the set, right? Two sets of chromosomes, one set from father and one set from mother. Or if we look at each type of chromosome, like chromosome number 1 from father, chromosome number 1 from mother, then we can say that in the diploid cell, the chromosomes are paired, or there are two copies of each chromosome. Okay, in a diploid cell, there are two copies of each type of chromosome, which we call the homologous chromosomes, right? A pair of homologous chromosomes. Now, somatic cells here means the normal body cells excluding the gametes. Okay, so normal body cells are all the cells involved in other processes, but not in reproduction. All right, like the red blood cells, nerve cells, and intestinal cells. So mitosis is involved in the production of all these different types of somatic cells, which make up a large amount of the cells in the human body. And then for meiosis, it's more sort of exclusive. It's only to produce gametes, haploid gametes. So a haploid cell, which we label as N, meaning one set. You see, the one is not written, so it's just an N. It means only one set of chromosomes. There's only 23 chromosomes, okay, if you're referring to the human haploid cell. So it is a cell that's only one set of chromosomes, only 23 chromosomes of all of different types, carrying different types of genes. So in meiosis, haploid gametes are produced, and examples are sperm in males and ovum in females. So meiosis aims to only produce gametes which have half the number of chromosomes compared to the normal somatic cells, right? And this is for the process of sexual reproduction, which we'll discuss later. Let's go on to the third difference. That is, what is the purpose of mitosis and meiosis? Purpose of mitosis is to produce new body cells through cell division for growth. Number 2, to replace dead, damaged cells. And number 3, for asexual reproduction in certain organisms like Hydra species. Firstly, mitosis produces new body cells for growth. For example, when a baby grows to become a child and then a teenager and then an adult, the body size increases because cell division by mitosis occurs to produce new cells to increase the size of the organism. Secondly, mitosis produces new cells to replace dead, damaged cells. For example, in the skin. In this layer of the skin, the last layer of the epidermis, which is called the Malpighian layer, mitosis occurs very actively, and cell division, that means cell division occurs actively to produce new cells to replace the dead and damaged cells that are shed from the skin, that drop off from the upper layer of the skin. Then, for certain organisms like Hydra species, mitosis occurs to produce new individuals from the parent, right? So these individuals are produced through mitosis, and it is a form of asexual reproduction in this case, because in asexual reproduction, only one parent is involved, as can be seen here. Only one parent is involved to produce the offspring, and there are no gametes involved and no fertilization is involved, right? So this is a form of asexual reproduction, and the offspring or the organism, the young organism that is formed, is genetically like the parent. Now, for meiosis, what is the purpose of meiosis? It's to produce reproductive cells. Textbook use the word reproductive cells, called gametes, that is the sperm for males in males and ovum in females, for the purpose of sexual reproduction. So through meiosis, the sperm and the ovum is formed. After that, these two cells will take part in sexual reproduction, whereby the sperm will fertilize the egg during the process of fertilization. So the nucleus of the sperm will fuse with the nucleus of the egg or the ovum. And then a zygote will be formed that will undergo growth to become a child or an offspring of the parents. So this is called sexual reproduction because two parents are involved, father and mother, not one, but two parents are involved, and fertilization is involved, gametes are also involved. So this is called sexual reproduction as opposed to asexual reproduction, in which mitosis is involved. So meiosis produces the gametes, which are haploid, having half the number of chromosomes compared to the parent cell, for the purpose of sexual reproduction, for the purpose of fertilization to produce the new offspring. Next, for difference number 4, let's discuss the number of cell divisions in mitosis and meiosis. Now, for mitosis, it involves only one cell division. That is, after the 4 stages, prophase, metaphase, anaphase, and telophase, PMET. Then comes the cell division, which is the cytoplasmic division, which is cytokinesis, right? And then 2 daughter cells are formed. So it's this part here that we are discussing. All right. So mitosis occurs, and after that, one cell division occurs. It could run almost the same time as the process of mitosis, right? So, but for meiosis, it involves 2 cell divisions because you have meiosis 1, where one cell divides into 2, and then there's meiosis 2, where each cell divides again into 2 cells. So in the end, there are 4 daughter cells formed, all right? So meiosis has 8 stages, PMET 1 and PMET 2. But I think you don't have to write this down. In the exam, you just write that mitosis involves 1 cell division, whereas meiosis involves 2 cell divisions. A very easy difference to give, right? Now, as for the other part at the top here, just let me explain. It's the same for mitosis and meiosis here. First, we have a parent cell at the beginning of the cell cycle. Okay, then it duplicates in S phase, so each chromosome becomes X-shaped with 2 sister chromatids each, right? And here we can say 2n equals to 2 for this cell. Why? Because it has 2 chromosomes that are alike, of the same length, so they are homologous chromosomes, and the black one could be from the male parent, white one is from the female parent. Okay, so we say there are 2 sets of chromosomes, although for each set there's only 1 chromosome. Okay, now so there's the black set and the white set. Okay, so 2 sets of chromosomes. So since this is a diploid cell with 2 sets of chromosomes, or 2 copies of each type of chromosome, so we use the term 2n to denote that it's a diploid cell, equals to 2. All right, because there are 2 chromosomes. Here 2n also still equals to 2, although the chromosomes have duplicated. All right, but we don't consider it as 4 chromosomes. So since it's still attached at the centromere, it's considered this one is considered 1 whole chromosome, and this one another chromosome. So mitosis occurs here, and then there's 1 cell division here, right? Now for meiosis involves 2 cell divisions, so we can see here in meiosis 1, the cell divides. After meiosis 1, the cell divides into 2, and after meiosis 2, the cell divides again. Each cell divides again to form 4 daughter cells, right? The 5th difference involves the number of daughter cells formed from 1 parent cell. So for mitosis, it produces 2 daughter cells from 1 parent cell. So if you look at this picture, starting from the second diagram, this is the process of mitosis. You can see that there's only 1 cell division right? After anaphase or telophase. So when the cell divides once through cytoplasmic division, 2 daughter cells are formed. Now, in meiosis, 4 daughter cells are produced from 1 parent cell. Since there are 2 cell divisions, right? Firstly, after meiosis 1 occurs, the cell divides into 2. Then, secondly, after meiosis 2, each cell divides again to form 2 daughter cells. So in the end, 4 daughter cells are produced from 1 parent cell, since there are 2 cell divisions. Difference number 6 involves the number of chromosomes of each daughter cell. Is it the same as the parent cell or different? For mitosis, the number of chromosomes of each daughter cell is the same as that of the parent cell. For example, in humans, if the parent cell has 46 chromosomes, at the end of mitosis, the daughter cell will also have 46 chromosomes, right? Or take, for example, a cell where the number of chromosomes are only 2. There are only 2 chromosomes in the cell, all right? So 2n equals to 2, because this is a diploid cell where there are 2 copies of this chromosome, the chromosome of this length. There are 2 copies, and 1 copy is from the male parent, is the paternal chromosome, and the other copy is from the female parent, which is the maternal chromosome. So here's a parent cell in interphase, and during the S phase of interphase, the chromosomes duplicate, right? So in mitosis, the chromosomes will first of all be arranged at the equatorial plane in metaphase. After that, the sister chromatids will separate from each other and be put to opposite poles of the cell. So each cell will have 1 sister chromatid for each chromosome, okay, from each chromosome. So in the end, the daughter cells will be exactly like the parent cell, because each daughter cell receives 1 sister chromatid, right, for each chromosome. So the number of chromosomes of the daughter cell, as you can see here, okay, is 2, just like for the parent cell here, okay? And then the daughter cells are also diploid, meaning to say there are also 2 copies of each type of chromosome in the cell. Or we can put it in another way, there are 2 sets of chromosomes. One set is the paternal chromosome, the black set, and one set is the maternal chromosome, denoted here by the white chromosome, okay? Now for meiosis, the number of chromosomes of each daughter cell is only half that of the parent cell. So if we talk about human cells, if the parent cell is a human cell, then it starts off with 46 chromosomes, but at the end of meiosis, the cells formed, which are actually the gametes, the daughter cells formed, which are the gametes, will have only 23 chromosomes, half that of the parent cell. So that's why meiosis is called reduction division. Reduction division, because in meiosis, in this type of cell division, meiosis, the daughter cells have only half the number of chromosomes compared to the parent cell. The number of chromosomes have been reduced to half, okay, that of the parent cell. So as we can see here, starting off with a cell that is that has 2 chromosomes, like for mitosis, how is the number of chromosomes halved? You see, in meiosis 1, homologous chromosomes will separate from each other. So this will cause the number of chromosomes in each daughter cell here to be half that of the parent cell already, because this is considered 1 chromosome, right? Not 2, 1 chromosome made up of 2 sister chromatids. It's also 1 chromosome in the cell, as compared to the parent cell, which has 2 chromosomes. Anyway, after meiosis 2, the sister chromatids would have separated, and each chromatid becomes a new chromosome, right? So there's only 1 chromosome in each cell, compared to the original parent cell, which has 2 chromosomes. So the number of chromosomes has been halved, has been reduced to half, okay, for the daughter cells, in the daughter cells. compared to the parent cells. So basically, you don't have to draw these drawings. You just write the difference as stated here. That's all. Okay. Difference number 7. For mitosis, the daughter cells formed are genetically identical to the parent cell. But for meiosis or in meiosis, the daughter cells are genetically different from each other and from the parent cell. Okay, so how is that so? Let's start with mitosis. Why are the daughter cells genetically identical to the parent cell? If you look at the parent cell in interphase, during the S phase, right, chromosomes duplicate. So from one strand, the chromosome forms another strand. Now, the original strand of chromosome or DNA, the original strand of DNA, and the strand that is formed, okay, when the DNA replicates, these two strands are genetically identical to each other. All right, 100% identical. So when, let's say you look at this chromosome, during Anaphase of mitosis, the two sister chromatids that are genetically identical separate and move to opposite poles. Therefore, the cell will have the same type of chromosomes, which are genetically identical to each other. Okay, because when chromosomes duplicate in S phase, the two sister chromatids formed are genetically identical to each other. However, in meiosis, the daughter cells formed are genetically different from each other and from the parent cell. That is because a process occurs in meiosis 1, prophase 1, called crossing over, where the non-sister chromatids of homologous chromosomes, that means the chromatids from the paternal chromosome and the chromatid from the maternal chromosome, they will touch each other and they will exchange genetic material, all right, through crossing over. Hence, after crossing over is completed, the chromosomes, okay, that are produced will have some parts from the other chromosome, and the genes may be different, all right, compared to the original strand. So in the end, when the homologous chromosomes separate and the sister chromatids separate, we will get the gametes formed, which are the daughter cells, that are genetically different from each other due to the process of crossing over. Okay, so there are new combination of genes in this chromosome, new combination of genes in this chromosome, okay, which is different from the other two. Difference number 8. Mitosis does not cause variation to occur, but meiosis causes variation to occur. Variation means the differences among individuals of the same species. For example, even in a family, there are differences in the type of hair and the color of the hair, eye color, face shape, blood group, and so forth. So all these differences are called variation. All right. Now, mitosis. Let's start mitosis first. It does not cause variation to occur. So if the offspring or the children, the anak, is formed through mitosis, they are genetically like the parents. So there are no differences. So there's no variation. All right. For example, this plant produces new offspring or plantlets through mitosis, through the cell division called mitosis. So the offspring or the anak are just exactly like the parent, genetically identical to the parent, because they are produced through the cell division called mitosis. Same for the Hydra species. For the parent and the offspring are genetically identical to each other because the offspring is produced through cell division by mitosis. Now, For meiosis, it causes variation to occur. Why? Because in meiosis, gametes that are formed through meiosis are all genetically different due to the process of crossing over and the random arrangement of homologous chromosomes at the equatorial plane during metaphase 1 of meiosis. Alright? So we have already discussed this when we discussed the process of meiosis. Alright? If not, just remember, because of crossing over, gametes formed are genetically different from each other, although they may come from the same parent. So when these gametes undergo fertilization, they'll produce individuals with different traits or characteristics. Okay, because the gametes were genetically different from each other. Now here for cats also, there is variation, because cats also, the cells undergo meiosis to form the gametes, the male and female gametes, which are genetically different from each other. Okay, so there is variation in the fur color, like a black or gray fur compared to orange fur, and also in the pattern that is produced on the fur. So now we're going to look more specifically at the phases of mitosis and meiosis to compare the phases. We'll start with prophase. So in prophase of mitosis, synapsis and crossing over does not occur at all. Here we see a cell in prophase where 2n equals to 4. That means this is a diploid cell with 4 chromosomes inside. Okay, 1, 2, 3, 4. So in this cell, we can see that the chromosomes do not carry out crossing over. Okay, you can just see the nuclear membrane here disappearing and the centrioles moving to the opposite poles. So in meiosis, during prophase 1, 2 processes occur. First, there's synapsis and then crossing over. Now, in synapsis, homologous chromosomes pair up to form bivalents or tetrads. Okay, this means that chromosomes of the same length carrying the same type of genes, one paternal chromosome and one maternal chromosome, pair up, they come side by side to form bivalents. Bivalents means 2 homologous chromosomes side by side, that are side by side. Or they form tetrads, because a tetrad consists of 4 chromatids. Okay, 2 sister chromatids from one chromosome and 2 sister chromatids from the other chromosome. So these two mean the same thing, bivalents and tetrads. Just call bivalents, I mentioned as bivalents, it's good enough. Now, that's synapsis, and then after synapsis, after the pairing up of the homologous chromosomes, crossing over occurs, where non-identical chromatids or non-sister chromatids of homologous chromosomes exchange genetic material. Non-sister chromatids means the chromatids from different chromosomes. One chromatid from the paternal chromosome carry out crossing over with one chromatid from the maternal chromosome. Alright? So this is to exchange genetic material in order to produce genetically different gametes. Okay, at the end of each cell division. And this will finally lead to variation, which is very important for the survival of the organism. Okay, variation is very important to occur to help the organism adapt to changing conditions in the environment and also for the survival of the organism. Now let's discuss the second phase, which is metaphase. So for mitosis, in metaphase, chromosomes are aligned or arranged in a single row or a single file at the equatorial plane in the center of the cell, and they are not paired. So it's like, your teacher asking you to line up in a single file, one after the other, right? One behind the other in a single file, maybe for assembly, all right? So the chromosomes here are not paired. And this cell is a diploid cell, meaning to say there are 2 sets of chromosomes, the blue set, maybe the paternal chromosomes, and the red set, maybe the maternal chromosomes, right? Another way of looking at it is that in a diploid cell, you have 2 copies of each type of chromosome. 2 copies of the long chromosome, 2 copies of the short chromosome. So this is a diploid cell. Now let's look at metaphase 1 of meiosis. So in metaphase 1 of meiosis, homologous chromosomes are arranged side by side in pairs at the equatorial plane in the center of the cell. So this is very different from metaphase of mitosis. Now in metaphase 1, it's like asking students to line up in pairs, right? One after the other. So there are 2 students together, side by side. 2 students together, side by side, right? So this is how the homologous chromosomes are arranged, side by side in metaphase 1, because the chromosomes will then move to opposite poles. And the chromosomal number, the number of chromosomes in the cell will be halved after that. So homologous chromosomes are arranged side by side so that they can separate from one another in anaphase 1, right? So take note now that when you see it like this, 2 chromosomes side by side, you know it's metaphase 1, not metaphase, right? Now, how about metaphase 2? So normally, if you are going to compare mitosis and meiosis, it's enough to compare metaphase and metaphase 1. All right? Here I add on metaphase 2 because sometimes in the objective questions, you may get a cell that looks like that, and you may have to identify what stage or what phase this cell is in, right? So how to differentiate metaphase 2 from metaphase, especially? These two look quite alike. Now, in metaphase 2, we can state that chromosomes are arranged or aligned on the equatorial plane. It's also like a single file, just like metaphase. But there's one big difference. The difference is that for metaphase 2, the cell is a haploid cell. Okay? It's a haploid cell. Whereas for metaphase, the cell is a diploid cell. Okay? So how to differentiate? Now you look at metaphase 2. You have only 1 copy of the long chromosome and 1 copy of the short chromosome. So this is a haploid cell. Okay? Because for diploid cells, you should have 2 copies of each type of chromosome. Okay? So here there's only 1 copy of the long chromosome, 1 copy of the short chromosome. So it's a haploid cell. It must be metaphase 2. So for meiosis 2, the cells are in haploid condition, especially for, that is for prophase 2, metaphase 2, and then in anaphase 2, of course, the sister chromatids would have separated already. So for prophase 2 and metaphase 2, the cell is in haploid condition. All right? And that's how you differentiate metaphase 2 from the other phases like metaphase 1 or metaphase. The last stage that we are going to compare is anaphase with anaphase 1. So in anaphase, in mitosis, sister chromatids separate and are pulled apart to opposite poles of the cell. And if you were to reverse the cell to metaphase stage, you'll find that it was a diploid cell. Okay? In this stage, we cannot ascertain whether the cell is diploid or not, right now. Okay? You should go back to the metaphase stage. Okay? And then join back, like play a movie in reverse. Join back the chromatids. You find that the cell is a diploid cell with 2 copies of the long chromosome, 2 copies of the short chromosome. All right? So then we know that this is anaphase of mitosis. Now you can see here that during anaphase, the centromere has divided and each chromatid is pulled to the pole, okay, with the centromere leading the way, pointing to the pole, like this. And then for each chromatid, you see it has sort of 1 arm and 1 leg only, right? Okay, now, for anaphase 1, it's not the sister chromatids that separate. It's the homologous chromosomes that separate. The whole chromosome will separate from the other copy, and they are pulled to opposite poles of the cell. the sister chromatids are still attached at the centromere, like these two sister chromatids still attached at the centromere. And same for this also. The two sister chromatids are still attached at the centromere. The centromere did not divide at all. Okay, so this is Anaphase 1. It follows from Metaphase 1. Okay, and this whole cell, if you look at this whole cell, it is a diploid cell because there are 2 copies of the short chromosome, 2 copies of the long chromosome. All right, so remember this difference. In Anaphase, the sister chromatids separate, but in Anaphase 1, it's the homologous chromosomes that separate and are put to opposite poles of the cell. Now, how about Anaphase 2? So normally, if you can differentiate between Anaphase and Anaphase 1 by writing all this down and differentiating between these cells, is good enough. However, for certain questions, they may give you Anaphase 2 and you may need to identify it, okay, especially for the objective section. So it's good to know how to identify Anaphase 2, right? Now, in Anaphase 2, the explanation is similar to Anaphase. Sister chromatids separate and are put to opposite poles, similar to Anaphase, right? Only thing is, this cell, if you reverse it back to Metaphase 2, you can see that it is a haploid cell. Okay, reverse back, play a movie in your head, reverse back to Metaphase. So these two sister chromatids will join back together. We find that it forms a long chromosome. And this one forms the shorter chromosome. So there's only 1 copy of the long chromosome, 1 copy of the short chromosome. So it's a haploid cell. Okay? Yeah, we don't have 2 copies of the long chromosome or 2 copies of the short chromosome. Okay? So it is not 2 copies, but 1 copy of each type of chromosome, so it's a haploid cell. Whereas in Anaphase, if you reverse back to the metaphase stage, you can see that it is a diploid cell with 2 copies of the long chromosome, 2 copies of the short chromosome. So here are the smart notes for what we discussed just now about the differences. For similarities, please refer to the beginning of this video, alright, and copy directly from the slide. So all the differences here have been compiled, and you can pause the video and then copy down the smart notes, or you can print it up too also. Next, there's one more part of the smart notes. Alright, this is the second half of the smart notes. So you can pause the video and then copy it, or you can print out this part of the smart notes. Alright, so before we end, let's train our brain and test ourselves by answering some questions on mitosis and meiosis. So let's test our understanding by trying some questions related to formative test 6.3. So the first question is, what is the difference in the number of chromosomes between a cell in meiosis 1 and a cell of the same organism in meiosis 2? Okay, difference in number of chromosomes. Meiosis 1 and meiosis 2. So is there a difference in number of chromosomes? Yes. The cell in meiosis 1 is a diploid cell, but the cell in meiosis 2 is a haploid cell. So this means that the cell in meiosis 1 has twice the number of chromosomes compared to the cell in meiosis 2, right? So for humans, the cell in meiosis 1 will have 46 chromosomes, but the cell in meiosis 2 will have 23 chromosomes only. Now, why does this occur? Why is there a difference in the number of chromosomes? Okay, for cell in meiosis 1 and cell in meiosis 2. Why is there a difference? How did it happen? Okay, let's come to the second question. Explain how meiosis 1 reduces the number of chromosomes in the daughter cell. Okay, so it answers that question. Now, meiosis 1 reduces the number of chromosomes in the daughter cells during Anaphase 1, when the homologous chromosomes separate and move to opposite poles, as seen here. Okay, so at first, if you look at this cell, there are 4 chromosomes. Okay, each chromosome is made up of 2 sister chromatids joined at the centromere. Now, when Anaphase 1 occurs, the homologous chromosomes separate from each other and move to opposite poles. So the new cell, imagine a line drawn in the center here. Okay, the cell will divide into two daughter cells that are intermediate before meiosis 2, before the end of meiosis 2. So anyway, if you look at the cells, look at this cell, it has now one longer chromosome and one short chromosome. Okay, so it is already a haploid cell. The number of chromosomes has been halved to 2, one long chromosome and one short chromosome, compared to the original diploid cell that has 4 chromosomes. Okay, same for this side, for this pole here, and for this the cell formed on this side, it will have one short chromosome and one longer chromosome. So there's only one copy of the long chromosome and one copy of the short chromosome in the new intermediate cell that's formed. Okay, so meiosis reduces the number of chromosomes in the daughter cells during Anaphase 1 because the homologous chromosomes separate and move to opposite poles of the cell. Here's another common SPM question. State one difference between metaphase of mitosis and metaphase 1 of meiosis. 1 mark. So here's our answer. In metaphase of mitosis, chromosomes are arranged or aligned in a single row like this, a single file or single row at the equatorial plane. That's according to the textbook, arranged in a single row at the equatorial plane, and they are not paired. Whereas in metaphase 1 of meiosis, homologous chromosomes are arranged side by side like this. So 2 homologous chromosomes together side by side. So homologous chromosomes are arranged side by side in pairs at the equatorial plane in the center of the cell. Alright, so this is how we describe the behavior of the chromosomes in metaphase of mitosis and metaphase 1 of meiosis. So you need to memorize this difference also. It's a popular question. Another question related to summative test 6. And this is also a past year SPM question. Alright, so it's important to know how to answer it. State one difference between anaphase of mitosis and anaphase 1 of meiosis. So we have discussed this in the earlier slide. Now let's look at the answer. In anaphase of mitosis, the sister chromatids separate and are pulled apart to opposite poles of the cell. But in anaphase 1 of meiosis, homologous chromosomes separate from each other and are pulled apart to the opposite poles. Alright, the sister chromatids still remain together and the centromere is still intact. It didn't divide, right? It's the homologous chromosomes that separate from each other. Okay, this copy of the short chromosome from this copy of the short chromosome. Same for the long chromosome too. This long chromosome separates from the other copy of the long chromosome. So try and memorize this difference, right? For anaphase of mitosis is the sister chromatids that separate from each other and are pulled apart to opposite poles of the cell. But for anaphase 1 of meiosis, homologous chromosomes separate from each other and are pulled to the opposite poles of the cell. Let's look at the last question here, which is a HOTS question and it's also a past year SPM question. Okay, so it's also found in your textbook. So it's a good question to try. Number 4, diagram 2 shows a cell at stage X in the cell cycle. Okay, so this diagram 2, spindle fibers do not form and attach to P. Okay, P is the chromosome. The sister chromatids of P do not separate. Take note of that. Draw the 2 daughter cells that are formed as a result of this occurrence. 2 marks. Okay, so 1 mark for each cell that you draw. So first of all, you have to determine what type of cell division this is and also what is the phase that the cell is undergoing. Okay, now first of all, find out whether it's a diploid or haploid cell. So you can see here there are 2 copies of the long chromosome, 2 copies of the short chromosome, right? So this is a diploid cell. Or you can say that there are 2 sets of chromosomes. One is the blue set and the other one is the red set of chromosomes, right? So this is a diploid cell and the chromosomes are arranged at the metaphase plane, or we say the chromosomes are arranged at the equatorial plane. Okay, textbook use the word equatorial plane, not metaphase plane. So stick to equatorial plane, it's better. So chromosomes are arranged at the equatorial plane in a single row or file. So this is a diploid cell. So this phase is actually metaphase of mitosis. All right. But here there's an abnormality. You can see that spindle fibers do not form at the top here to pull the sister chromatids apart at anaphase, during anaphase, right? So now you are asked to draw the 2 daughter cells that are formed as a result of this abnormal occurrence. Okay, so you have to predict the result. To predict the result, use your imagination to imagine anaphase. What happens in anaphase? In anaphase, the sister chromatids will separate from each other and move to opposite poles. So it will occur for these 3 chromosomes. All right, sister chromatids will separate and move to opposite poles. But for the last chromosome, it's not the same. For the last chromosome, the 2 sister chromatids will be pulled to the pole below here. And there's no P pulled to, or no sister chromatid of P pulled to the upper pole, right? So by now you should be able to predict what the 2 daughter cells will look like. So try and draw the 2 daughter cells that are formed. Okay? So pause the video for a while, and then after that we can discuss the answer. Okay, let's look at the answer. Now let's imagine the daughter cell formed when the sister chromatids move to the lower pole here. Okay, so you will have 1 long red chromosome here, 1 short blue chromosome here. Actually, it's the sister chromatid, right, that moves to the pole, and each sister chromatid becomes a new chromosome. All right, and then 1 sister chromatid which is short and red. Okay, like this. But for P, the whole chromosome will move to the lower pole. Okay, so you draw the whole chromosome, and the centromere did not divide, the sister chromatids did not separate from each other. So it looked like that. Okay, as it is here. For the other cell, we can imagine that the sister chromatids will be pulled to this pole here for these 3 chromatids. 3 chromatids will move to the same pole, right? So you have 1 red short sister chromatid which will become a chromosome, and then the blue short sister chromatid, and then the red long sister chromatid. Okay, so only 3 chromosomes are found in this daughter cell, and this one has extra bit of this chromosome. Okay, has 1 extra DNA strand here for this chromosome. All right, so I hope you have understood how to draw the drawings. Thanks for viewing, and please do share, like, and subscribe. So till we meet again, goodbye.